Welcome back to Chapter 10, Part 2, focusing on Section 10.2, the structure and function of DNA. In this section, we'll review DNA's building blocks, its double helix structure, and how these features relate to DNA's role as the hereditary molecule. DNA is built from deoxyribonucleotides, and each of these deoxyribonucleotides is composed of 3 parts, including 1, a deoxyribose sugar, 2, a phosphate group, and 3, a nitrogenous base. The nitrogenous bases include A, G, C, and T for DNA, and these stand for adenine, guanine, cytosine, and thymine. If you remove the phosphate group, what you have left over is just the sugar and base, and in this case we call these nucleosides. So with the phosphate, they're nucleotides. Without the phosphate, they are nucleosides. And for the nucleotides, we often name them by the base that is attached to them. So here we have deoxy, and this is going to be ATP, GTP, and I'll tell you guys more about these in a bit. So here's a diagram of our deoxyribonucleotide with those 3 components. Nucleotides have all 3 components. We have our deoxyribose sugar, our nitrogenous base, A, T, G, or C, and our phosphate group. If we're just looking at these components here, the sugar and the base, remember that would be a nucleoside instead of a nucleotide. If I look more closely at my deoxyribose sugar, here we have a difference between DNA and RNA. We'll see later that there is no hydroxyl group here. In RNA, which we'll see in the next video, we're going to see that there is an OH or hydroxyl group at carbon number 2. It is important to know the numbers of the carbons within the deoxyribose sugar, and they are numbered as in this diagram. So carbon number 1 is always the carbon that is attached to the nitrogenous base. So here's carbon number 1 indicated by this vertex or this point. Carbon number 2, 3, 4, and this is the 5th carbon. We'll see why this is important in a bit. And in the previous slide, I mentioned they are named. Each of these deoxyribonucleotides is named after their nitrogenous base. DATP, for example, stands for deoxyadenosine triphosphate, and we'll see later why there are actually initially 3 phosphate groups attached. DNA bases, those nitrogenous bases, fall into 2 categories. Purines include adenine and guanine and have a double ring structure. So you can see these have 2 rings within each of the structures. Pyrimidines, in contrast, only have a single ring in their structure, and these include cytosine, thymine, as well as uracil. It's not shown in here, but uracil is also a pyrimidine. When I was learning this, my professor taught me to remember these categories by thinking about pure silver. The symbol for silver is Ag, and then pyramids, or pyrimidine, kind of sounds like pyramids, and pyramids are kind of sharp and pointy, so pyramids cut. Cytosine, uracil, and thymine are pyrimidines. We'll see later that during base pairing in double-stranded DNA, purines will pair with pyrimidines, and we know that thymine is unique to DNA, whereas uracil is found in RNA in place of thymine. DNA strands are made of monomers called nucleotides or deoxyribonucleotides, and the bonds connecting these monomers are phosphodiester bonds, more specifically 5' to 3' phosphodiester bonds, where the phosphate on the 5' carbon of one nucleotide joins the 3' hydroxyl on the next. This creates a sugar phosphate backbone with 2 distinct ends, the 5' end carrying a free phosphate and the 3' end carrying a free hydroxyl group. DNA synthesis and reading, we'll learn later on, occurs in the 5' to 3' direction, and this will be important when we learn more about DNA replication as well as transcription. Here's a closer look at part of a DNA strand. I have 1, 2, 3 nucleotides here. These are all deoxyribonucleotides, and I can see that phosphodiester bonds are what connect one nucleotide to the next. So here's my 5' end, that 5 with a little apostrophe there, that is the prime, that's known as prime. And my second nucleotide over here connects to the first nucleotide, 1, 2, by an ester bond here. And this one is already connected, this phosphate group is already connected to this sugar by an ester bond. That's why this is called a phosphodiester bond that connects these monomers. If I look, the 5' end is connected to a phosphate group over here, and the 3' end on the other side, or the third nucleotide, has a hydroxyl group. I also wanted to talk about how earlier I mentioned that the nucleotides are named after the base. So this would be DTTP, here would be DCTP, and DGTP. All right, so the T in the middle stands for triphosphate, but at the end you can see there's only a single phosphate here. Only one here, only one here. So what happened was, to form DNA, as the strand is getting longer and longer, the other two phosphate groups are released. So originally it was 3, and the release or the breaking of these bonds releases energy to power the elongation or formation of DNA. So what's meant by the 5' and 3' ends of a nucleic acid strand, we're referring to carbons on that deoxyribose sugar. So each carbon is numbered. Remember, 1' is the one that's attached to the nitrogenous base, and then 5' is the one that is attached to the phosphate group. So the 5' end of a strand always has a free phosphate group on the 5' carbon. On the other end, so the opposite end of that DNA strand, we're going to always have a 3' end with a free hydroxyl group, that OH group, on the 3' carbon. We're going to learn later on that DNA and RNA are synthesized and read in the 5' to 3' direction. So if I wanted to add another nucleotide, I can only do so on the 3' end. You cannot add or grow from the 5' end, only the 3' end. Erwin Chargaff studied DNA-based composition and found that the number of A's within a DNA molecule is always very close to the number of T's, and the same thing is true between G's and C's. And this is true within each species, but the ratios can vary between species. For instance, within a single species, they might have 60% G's and C's in their DNA, but only 40% A's and T's. So this is maybe species 1. But in a second species, a completely different species, maybe they have only 40% G's and C's and 60% A's and T's. So again, within the same species, they would have similar makeup of DNA bases, but between two different species, they could have very different ratios. And these relationships really provided key evidence for the complementary base pairing. Now we know that A pairs with T and G pairs with C within DNA structure. Rosalind Franklin and her student Gosling used X-ray diffraction to show that DNA is helical in structure. It's a repeating structure with the DNA bases inside. Remember, the bases are the A, T, G, C. And on the outside, as part of the backbone of the helix, we have our sugar phosphate backbone. This famous photo really gave essential clues to DNA's actual shape. And using Franklin's data as well as Chargaff's rules, James Watson and Francis Crick proposed the double helix model of DNA in 1953. This is actually a picture of the model from a display at the Science Museum in London. So DNA consists of two strands that are anti-parallel. One strand will run from 5 to 3', whereas the other strand will be from 3 to 5. They'll be facing in opposite directions. The sugar phosphate backbones are on the outside, and the bases will be paired inside by hydrogen bonds. A is going to be paired with T with 2 hydrogen bonds between them, and G and C will be paired with 3 hydrogen bonds between them. There will be about 10 base pairs per turn of the helix. So in this picture, I can see that the strands are anti-parallel. They're running from 5 to 3 on one strand and 3 to 5 on the other strand. They're facing the opposite directions. I can see the 2 hydrogen bonds between A and T, 3 between G and C, and I can see that the backbone is the sugar phosphate backbone, while the bases make up the rungs of the ladder, right? They're in the center of the helix. When you look at the number of base pairs per turn of the helix, what that means is, let's just pick any arbitrary point. I'll pick this point of the helix, and if I get all the way back to the same direction, so from about here to here, that is going to be about 10 base pairs that fit per turn of the helix. So the scientists that are given most of the credit for describing DNA, most people think of Watson and Crick because they proposed that double helix model in 1953. But remember, they built on previous scientists' data, just like the X-ray diffraction data from Rosalind Franklin and R.G. Gosling. But Watson and Crick, ultimately, they combined all of that information to define that anti-parallel double helix structure with complementary base pairing. Going back to that image of the double helix, there's actually unequal spacing of the backbones. So there are major grooves where there's a greater space between the backbone, as well as minor grooves, smaller spacing between the backbones. So the major and minor grooves are actually binding sites for proteins that regulate DNA replication and transcription. So we'll learn more about this later on. Proteins will need to bind to DNA, DNA, when they're going to replicate DNA before the cell divides, and transcription is when we're going to read DNA and produce messenger RNA. And here's a closer look at our base pair hydrogen bonding. A and T pairs have 2 hydrogen bonds, while C and G pairs have 3 hydrogen bonds. This makes CG-rich DNA more stable and harder to melt, and this difference will underlie variations in DNA's melting point among different organisms. Just like we saw in proteins, heat or chemicals can also denature DNA. That means the 2 strands would be separated. So double-stranded DNA would denature and form 2 single-stranded DNA molecules. But if you were to cool the DNA down or remove the chemicals, this would allow the strands to renature or re-anneal. That means you're reforming those hydrogen bonds between the base pairs. These properties are exploited in lab techniques like PCR, polymerase chain reaction, that we're going to learn more about later. And because G's and C's pair with 3 hydrogen bonds instead of 2, DNA with higher GC content will require higher temperatures to denature. So here's another look at that. Here's normal DNA, and if you add heat or chemicals, you can actually denature DNA, forming single-stranded molecules. If you cool it down or get rid of those chemicals, DNA can renature, reform those hydrogen bonds. Right, so what are the 2 complementary base pairs of DNA and how are they bonded together? Adenine, or A, pairs with thymine, T, and that occurs with 2 hydrogen bonds, whereas cytosine, C, pairs with guanine, G, through 3 hydrogen bonds. And notice that it is a purine and pyrimidine pairing, the single ring and the 2 ring. So single ring were pyrimidines like Cu or T, in this case just cytosine and thymine for DNA, and the purines were A and G with those 2 rings. So it's always a single ring pairing with a double ring in order to keep the helix, that double-stranded DNA helix, uniform in width. And the more G and C pairs you have, the more DNA will be stable and harder to denature. DNA structure allows it to store and faithfully transmit hereditary information. During vertical gene transfer, this information passes from a mother cell to daughter cells, so from one generation to the next. DNA can also direct RNA and protein synthesis, and we'll see this in a future chapter, and its nucleotides can be recycled. Unlike other macromolecules, DNA's main role is informational and not structural. So this will be a main topic for another chapter, but in general, how does DNA transmit genetic information to offspring? We're going to see that during DNA replication, each strand will serve as a template for a new complementary strand. And this process will copy the sequences of bases, preserving the genetic code. The copied DNA is going to be passed to daughter cells during cell division, and this will be part of the vertical gene transfer I talked about. Because base pairing is complementary, so A pairs with T and G pairs with C, the information will be faithfully transmitted to offspring. And at the end of this section, our book provides evidence of underrepresentation of women in STEM. Even today, globally, women make up only about 28% of the STEM workforce. and there are many reasons why underrepresentation persists. There is often something known as the leaky pipeline effect, where women are more likely to leave STEM at various stages. There's still implicit bias and structural barriers like stereotypes, unequal mentorship, fewer role models, and there are many studies in confidence and self-efficacy gaps. These studies show that women often report lower confidence in STEM, even when their performance is as good as, or maybe even better than their male counterparts. So overall, it's very important to be aware of any kind of stereotype or structural barriers for any underrepresented group, and I hope we can keep this in mind as we continue in the STEM fields. All right, let's recap 10.2. So if I look at the biochemical structure of these deoxyribonucleotides, they're made of three parts: the deoxyribose sugar, the phosphate group, and the nitrogenous base. So that sugar is deoxyribose. It's a 5-carbon sugar without an oxygen on the carbon number 2. The phosphate group is attached to that carbon number 5 of the sugar, and the nitrogenous base, A, G, C, or T, is attached to carbon number 1 of the sugar. And remember, these nucleotides are linked together by phosphodiester bonds to form the DNA strands. For the second one, the base pairs, so purines are adenine and guanine, and the pyrimidines in DNA are cytosine and thymine. We know that A pairs with T with 2 hydrogen bonds, and G pairs with C through 3 hydrogen bonds. These pairings ensure that the helix is uniform in terms of width, and we have accurate DNA replication. So the last one, why is it anti-parallel? Remember, DNA consists of 2 strands that are running in opposite directions. So one strand runs from 5 to 3 prime, the other one is 3 to 5 prime. That opposite orientation is called anti-parallel. And this anti-parallel arrangement really allows for a complementary base pairing, and we're going to see later on, enzyme binding during replication and transcription. All right, that takes us to the end of our second video. In the next part of Chapter 10, we'll review the structure and function of RNA as well as cellular genomes.